probably be the most interesting class that you're probably going to have right now, because he's working on something that's really, really cool. So I'd like to introduce Dr. Elazar Eskin, professor in computer science at UCLA. And Dr. Eskin directs the ZAR Lab, a computational genetics group affiliated with both computer science and also the human genetics department at UCLA, which is actually a first. Also, Dr. Eskin's research interests are around computational genetics and specifically focusing on developing and applying techniques from computer science, statistics, and also bioinformatics to understand the relationships between genetic variation and traits to understand the pathology of different human diseases. So I'd like to introduce Dr. Eskin.
Okay, thank you. So it's great to speak to you guys. Everybody here is a freshman in computer science, right? Right, yeah, and a required course. And they can make you do a quiz on whatever I'm going to tell you today, like on Wednesday, right? So that's why you all have to be here, okay? Well, I'm going to try to make this class, even though you have to be here, I'm going to try to make this class interesting, and I'm going to talk a little bit about, instead of what's inside computer science, which is what you'll learn in the bulk of your courses here, I'm going to talk a little bit about what computer science can do in other areas. And particularly, I'm going to be talking about like biology and medicine. But if you think in a little bit broader sense, really there's kind of grand challenges for our society. Two of them are human disease, and one's human disease, another one's energy. And really a lot of these now require a lot of data analysis. I mean, there's a huge amount of kind of data that is, needs to be analyzed for these things. And like the only way to analyze that data is with computers. And what I just realized right now is that this plug doesn't work, okay? And so we'll have this dramatic, like power outage. So is there any plugs that work up here? Huh? Yeah, I mean, do you think you can get like an extension cord or something? I mean, it probably lasts for like, yeah. Okay, yeah, if you have a longer one, that should work too. Okay. So, so what I'm going to be talking, so basically both of these, so only really computers can analyze this data, even though you can't see anything that's written down here. I know that because I've taught classes in this classroom, but this is what it says, only computers can analyze that much data. So what I'm going to tell you about is what my lab works on and my research group works on. And how many people have seen the movie Gattaca? Okay, so that's basically what my lab is doing, with the exception of the whole kind of love triangle thing. We fortunately or unfortunately don't have that. But in this world of Gattaca, this actually movie was about 97 or something like that, so it's pretty old now. But it had presented this future where, based on people's genetics, their whole lives were kind of predetermined and things like that. So we're working on that. This is a great movie. If you haven't seen it, I highly recommend it. There's actually a scene in there where like a woman, there's these clinics that test people's DNA and predict how good and smart they, their future. So there's this scene where this woman goes to one of these screening places because she kissed a guy, and like she has him take a test of like his slobber off of her lips to find out like if he's good enough of a person for her to marry, So these are the kinds of things that we'd like to predict. But but of course, even though we like to predict that, no one's going to really support that kind of research, but people support medical research that works on the same technology. So there's this idea called personalized medicine, okay? And that's really what the future of medical care is. And the basic idea is that if you think about it currently, you go to the doctor and you have some symptoms, and based on those symptoms and kind of statistics of what treatments have worked for people with those symptoms, they give you a treatment. And you can think of two possible categories of explanation of what those treatments are. Okay, one of them, like how that works. One of them, the treatment or the drug can be either toxic or not toxic. okay? And the other one is that the treatment can either be beneficial, effective, or not effective, okay? And each person kind of falls into these 4 categories, and the difficulty is that right now, the only way that a doctor can find out if it's going to work or not is by basically giving you the treatment. So what the idea behind personalized medicine is that maybe we can actually measure some kind of genetic factor, genetic variant that you possess, or some kind of a biomarker, maybe some kind of a level of some chemical in your blood that can help us predict into which category you'll fall for each of these treatments. So that way what we could do is we could give you a treatment that corresponds to that. Hey, listen, if I don't get that power... Okay, okay, sounds good. Okay. That'd be kind of embarrassing. Okay, anyway. Okay, so there's actually an example of a treatment. So warfarin, has anyone heard of the drug warfarin? Okay, so this is a drug that's a kind of a blood thinner that's very effective. So if you're, as you get older, or your parents, depending on, some people have heart conditions that give them a risk of stroke, and warfarin is basically a drug. Aspirin has the same kind of properties that basically thins your blood to make you less likely to have blood clots. So blood clots can cause heart attacks and strokes, and this is a very effective drug, okay? The only issue with it's very effective. The only difficulty with it from a medical perspective is that different individuals can process vastly different amounts of the drug. So some people's dose is 10 times what other people's dose are. That's like the range. So it's very hard for doctors to set a dose for an individual. What they typically do is they just start with a very small dose and they just keep on increasing it until they start to have like medical problems, like their blood's too thin, they start passing out. So obviously you can imagine that the only people that would take warfarin already have heart problems, and so this is a very difficult procedure in setting this dose and risky. The way that they actually discovered warfarin and these different doses is that warfarin used to be a rat poison, okay? And essentially some guy tried to kill himself by eating a lot of this warfarin. He ate an insane amount of rat poison, and he survived, okay? And the reason why he survived is because he had these genetic variants that affect how the liver actually processes warfarin and really breaks it down. So that's the reason why he was able to actually absorb a huge amount of warfarin. Yeah, that'd be perfect. Thank you. Okay, so so now the idea is that now that they know this, what they could do is they can actually test individuals to see if they have these variants. And actually, here there's a website, right, which you can actually go to. It's called warfarindosing.org if you ever have to take warfarin. And what you do is you basically plug in a lot of information about you, and then you also plug in a bunch of your genetic variants at these positions, and then it'll tell you what dose to start at. Okay, so that's an example of a personalized medicine. It's personalized because this is based on your own genetic kind of profile. And so this is similar to Gattaca, of course, not as exciting. For the kind of, there they try to predict if you can, fly spaceships and stuff like that. Here it's like what your starting dose is for a medicine, but it's like the same basic idea, right? Okay. So I'll give you a little bit of historical context on what's going on in this field and why things are exciting. So can anyone here recognize more than one person in the photo? Okay, can anyone here at least recognize one person in the photo? Okay, so the person in the middle, his name is Bill Clinton. He was a president probably when you were like in junior high or something like that. So these two guys here, and this is an event in 2000. It's kind of a big event in science. So this is a press conference, okay? And if you have a press conference where the president is speaking, that means that it has to be big news, okay? Even if it's incorrect. I think Bill Clinton had another press conference when they thought they discovered life on Mars, which actually they found out that it was fake, but he also spoke at that one too. So this was a big event. Essentially, these two guys here, this guy anyone know who this guy is? Okay, his name is Francis Collins. He is a really famous geneticist. He discovered cystic fibrosis gene. Anyone heard of cystic fibrosis, the disease, right? So he discovered that gene, and he kind of pioneered this kind of discovering genes of all disease, and then he led the Human Genome Project. People have heard of the Human Genome Project, right? Kind of, right? This guy here is his arch nemesis, this guy named Craig Venter. So Francis Collins was leading the public effort to sequence the human genome. Craig Venter was leading this company called Celera, which was trying to sequence the human genome also. And actually, they used to work together. Craig Venter was at the NIH, where there was this Human Genome Project, and they actually had an argument over which mathematical algorithm to take all the data that they generate and construct the human genome. And essentially, Craig Venter kind of lost that argument, and they decided to go with not the approach that he was advocating. So what Craig Venter did was he said, screw you guys, I quit. I'm starting my own company. I'm going to sequence the human genome, and you guys might as well sequence the mouse because we'll do it so much faster. And so anyways, because of this, there's this big tension, this big fight, and kind of they finished at the same time. So they had this press conference. So this is announcing the sequence of the human genome. And so they said a bunch of interesting things. One of the things that he says that they've reached this milestone covering the genome and kind of a working draft of the human genome. And then one of the things that they say is it's not that useful. And one of the things here is that Francis Collins says is that he says that this is in 2000. He says that within 10 years, what he predicts is that we'll be able to take someone's DNA and predict what diseases they're at risk for. And so that was obviously, it's now 2013, so it's more than 10 years. And so we can look a little bit at what the progress is. And they've actually made a lot of progress. So here's actually kind of a way to visualize how much progress they've made in disease. Okay, so in predicting disease. Okay, here are 4 diseases. I don't know if you guys can see, but these are this one is Alzheimer's disease, breast cancer, type 2 diabetes, and BMI, obesity. Okay, and there's 3 colored bars here, which are hard to see because this projector's colors are not right, but this bar here, which is supposed to be green, okay, it is the average risk in the population. So for example, you have about a 28% chance of getting type 2 diabetes just because that's the average risk, okay? So without knowing anything about a person, you talk to a person in their lifetime, are they likely to get diabetes? It's 28%, okay? These 2 bars here are kind of the extreme cases. So like this bar here is if you are extremely unlucky in the gene pool lottery, okay, and you have every genetic variant that increases your risk of having type 2 diabetes, okay? So if that's the case, you're going to have, it looks like about a 75% chance of having type 2 diabetes, right? And similarly, this one here is if you win the gene pool lottery and you have every variant that protects you from diabetes, it looks like instead of having a 28% chance, you'll have a 24% chance. Okay, so obviously these 2 bars are really showing the extreme cases, okay? But you can kind of, it's a way to visualize how much progress there is, right? So the farther the distance between these 2 bars and this middle bar, that kind of reflects how much we know. So the more variants we know, the higher this will go and the lower this will go. Okay? It also tells you something else. So for example, if you look at Alzheimer's disease, where you have, if you have a certain variant, you might have a very high risk. Okay? But if you don't have but like for type 2 diabetes, even if you have all the genetic variants protecting you, you still have almost the same risk as if you don't have that. So in some sense, we're like good at predicting who will get the disease, but we're not good at predicting who won't get the disease. Does that make sense? So you can kind of see that, right? This actual variant here is pretty scary. It's like a single variant, and it counts for a huge amount of disease. Okay, so the question is, and what we're going to talk about today is how do they find these? Okay, and then we'll get into some where the computer science is involved. Okay, so here's really, so here actually, so here is okay. Actually, you know, even before that, let me ask you a question. How do you think you could find genes that are involved in disease or variants that are involved in disease? Any ideas? Yeah. Huh? Trial and error. Okay. A little more specific. Trial and error of what? Yeah.
So maybe like take a pool of people who have the disease and then look at their gene pool and then see which genes they have that are in common.
Okay, so you could take a pool of people that have the disease and look for like genetic variation that they all share, right? That's a good idea. Okay, there's a that kind of works. There's a small difference, yeah. So a person, yeah. So that's exactly it. So you take a pool of individuals that have disease, pool of individuals that don't have the disease, and you compare the two. Okay, and so this actually all started very recently. So in 2007, which is not that long ago, this real process started. They're doing exactly these studies. So Science magazine it's a big kind of publication in science. It's at the end of the year, it lists like the breakthroughs of the year. In 2007, the big one was human genetic variation. And during this time, it was a big deal because they discovered 50 genes that were involved in dozens of common diseases like diabetes, cancer risk, Alzheimer's, et cetera, okay? And 50 at the time was a big number because previously they only had a very small handful. So if you think about 50 genes, that's almost a weekly news report of scientists discover gene causing blank. And so this is a big thing. This is a big news, okay? And again, how do they find these variants? So exactly what you said. So what they do is they take two populations. One is called the cases. These are individuals that have the disease. And one is called the controls, the background population. And what they do is they look for differences between these two populations to see if a gene is involved in disease. So let me just show an example. So here is an example of, let's say, DNA that comes from two individuals, two populations. So let's say each row here is the chromosome of an individual, okay? And so each of these columns is a specific position. And these are called cases, individuals with the disease, and these are controls, okay? So, of course, in my slides, it's actually colored, and here it's not really colored. But can anyone see the position that differs between these two populations? Yeah? Where's that?
There's a row of A and C's kind of off to the right of A's.
This one?
Yeah.
Okay, so how many A's and C's are here?
4 C's and 4 A's.
And how about here?
5 C's and 3 A's.
Yeah, so is that really different? Yeah. I faked this example, right? So, so anyone else, any other guesses? It's hard because it's kind of, yeah, yeah.
I don't know how to describe it, but there's much P's and C's.
Where? Yeah, there's another one actually. Yeah.
8 to the left of the original column is another column that goes from G to C.
Yeah. So here it's like, where is it? This one, right?
Yes, that one.
So it's almost all G's here and almost all T's here, okay? So in the slide, the different ones are colored, so it's actually easier to see, okay? So I don't blame you, even though you guys are the first class that hasn't gotten it in like 5 seconds, but I blame the projector and not you guys, right? So, okay? So yeah, if I highlight it, so this is what's called an associated variant, okay? Does that mean that this variant is causing the disease? No? Why could it not be that? Yeah, so correlation doesn't imply causation. That's like one of those general things that you can say and it's always correct. And pretty much always the answer to everything, right? But like more specifically, what else could be a reason? Yeah? Well, yeah, that's one thing. But for these, remember, these variants just increase risk. So it doesn't like for example, this might be cardiovascular disease, like heart disease, and this guy here is a marathon runner, you know, so he's like in super fit, or and you know, this guy here, like is you know, eats like how we know we're not supposed to eat, you know, for example, right? So just increasing in risk, right? So none of these things are 100% determinants. But what's another reason, like a more simple reason that this could not be the one? Yeah?
They're different people, so they probably have other genes. Right?
Well, yeah, they have other genes, but even still, like how many individuals is this? Huh? Well, it's like eight people in every group, right? So what could happen? Yeah?
There are just so many genes that you can happen to line up.
Yeah, exactly. So just by chance, right, you can see that this discrepancy, okay? Because of that, studies today have tens of thousands or hundreds of thousands of individuals, right? And they collect the whole genome, so they're collecting millions of variants. So you need like huge numbers of people in order to be able to rule out that it's not by chance, okay? Okay, so why are people doing these studies now? This is such an obvious idea, right? This is not like, you guys thought of this idea in like 2 minutes of hearing about it. So why are people doing it now? Well, the main reason why people are doing it now is because the cost of collecting DNA genetic variation is dropping really rapidly. So here actually is the cost where this is on the log scale, and this value here is the cost to collect 1 million DNA positions. Okay, so 1 million DNA, our genome is 3 billion. Okay, so that means that there's 3,000, right, of these millions. So you can imagine that this is really expensive, right? So 3,000 times this number is what? Huh? 3 billion, right? So 30 billion, right? So the cost when they started when they finished the Human Genome Project, when they started the Human Genome Project, they thought it would cost $3 billion to complete it. Okay, when they finished it, because technology improved, it cost about $1 billion total. Okay, and today though, it costs less than $1,000 to collect a genome. Okay, that's a huge decrease in cost. That's a decrease in 10 years of a factor of 1 million in terms of the cost. Okay, anyone here heard of Moore's law? Yeah. Okay, good, right? So Moore's law, this is also on a log scale. Okay, you can see that Moore's law decreases much slower. So the actual cost of the computing, right, is decreasing relative to the cost of collecting of the data collection, which means that if you're spending the same amount of money per year on collecting data and on analyzing the data, right, there's this big gap opens up, right, because you're actually collecting much more data over time, then your computers are getting faster to actually analyze it. Does that make sense? So that's actually why we need better algorithms, and it means job security for people like you and me. Okay. So what did they find from this? So here's an example. So this is actually what they found. This is a plot from 2009. Okay. These vertical lines are human chromosomes, and each of these circles is a dot. Each of these circles is a location that's implicated in a disease. Okay. So these are all the diseases, kind of the legend down here. So you see that there's many, there are many, this is, 2005 they had 50. They're clearly, here they have 439, okay? And then this is just 1 year later. It doubled again. Now they have, this is 2010, this is like 900 something. And then this is actually the most recent one they have, which is 2012. You can see that there's huge numbers. So they've found hundreds and hundreds or even thousands of genetic variants that are implicated in disease. Okay, so now I'm going to skip a little bit ahead. Okay, so now let's talk about kind of an active research problem, which is, kind of, so I've given you all this background, so let's talk a little bit about where computer science and where algorithms can fit in. Okay, so this is the problem that we're going to talk about. Well, it's called short read resequencing, and another way to think is where are my mutations? Okay, so let's say I get recruited because I have some weird disease, and I get recruited into a disease study where I'm going to be a case, and so they want to collect my DNA to put it into the study. Okay, so obviously, I had the DNA is inside me. Okay, so if I spit, okay, the DNA is in my spit. So the question is, how does it get converted into something that we can analyze, like to actually get the sequence? Okay, and so there's me. Okay, and then there's my DNA. Okay, and so that's what we want. And so we want to get from the molecules in my spit, we want to get my DNA, and then we also want to figure out where all the variants are, et cetera. And so what we're going to do is we're going to use these new sequencers, a machine called a sequencer, where essentially it takes the DNA molecule and spits out the sequence. And these sequencers, we're going to use one of these new sequencers. They're called next generation sequencers. They're the ones that really drove the cost down, and they're very inexpensive to use. 1,000,000 times cheaper than 1,000 Genome Projects. But they have this characterization which is called short reads. They're called short read sequencers. And I'll show you why they're called that. So basically, here's my full DNA sequence, but the sequencer doesn't give me my full DNA sequence, right? What the sequencer does is it generates kind of random short substrings of DNA of a certain length. So what I mean by that is this is what it does. Okay, so there's my full DNA sequence that's encoded in the molecule, okay, and the sequencer just spits out these reads, okay, these sequence reads. And one way you can, of course, the sequencer doesn't work like this, but imagine the sequencer inside knows the full DNA sequence, and what it does is it picks a number from 1 to 3 billion, okay, and whatever that number is, it just spits out maybe the sequence of the next 30 positions. But not what that number was. It doesn't output where that sequence started. So you just get these huge numbers, huge list of these 30 length substrings randomly selected from your full sequence. And so the goal is, how do we recover the original sequence? Okay? So so that's the problem. So here are some difficulties, right? So I don't know where each read came from. Okay, so all I get is a list of reads like this, and I can't identify where the variants are. So this isn't enough to do a study, right? Because I don't know, like if I want to compare my genetic variation to somebody else's, I can't really do it. So the question is, what do we do? So this is where I ask you guys, any ideas? Yeah. Yeah. So they're so in general, you collect maybe 10 times or 50 times more total sequence that you collect per position. So if your length is let's say length 3 billion, you're going to collect a total length of like 30 billion or 60 billion. So you're going to by chance you're going to have lots of every position is going to be on average collected many times. Okay, so that's a great idea. Okay, so the idea is this. Why don't we look for overlaps and then, do that. So you take two sequences of length 30, okay? And if they overlap, let's say by the last 10 positions, then you can extend that to a sequence of length 50, right? Then you can find another one that attaches there and extend that maybe to a sequence of length 70, 90, 110, 130, et cetera, and just keep on repeating that process until you get to 3 billion. Okay, it's a long time, but computers, you can leave the computer running over however long it takes and then do that. Okay, so will that method work? And will that work? No? So why won't it work? Yes. So some parts of the DNA might be the same. So that like sequence of length 10, it might match to many different other reads. Okay. First of all, you're actually collecting a lot more reads. You're collecting 10 times as much, so every position's in there. But even if you don't assume that, okay, let's say you just collect everything only once, it might match more than once. Okay. So now let's look. Why does it match more than once? Okay. Actually, even better. How many times will it match? Can anyone do this in their head? Okay, so let me walk you through this, okay? How many possible sequences are there of length of, ACGT, there's four, right? So it's how many are total? Huh? 30 to the fourth power, almost, yeah? 4 to the 30th power, right? But we're only looking for matches of length 10, right? So there's 4 to the 30th possible reads, agreed. But when we're looking for only that end position, right? How many? It's 4 to the 10th, right? How much is 4 to the 10th? Okay, it's a lot, right? Okay, more precisely, yeah? 2 million-ish? Okay, so listen, I'll give you guys a hint, okay? You have to know that, you have to be, anyone know? 4 to the 10th. Yeah?
1,048,570.
And you did that on your computer?
Yeah.
Okay, but how can you just know that right away? 2 to the 20th. And what is 2 to the 20th? Well, yeah, but how do you do that? 2 to the 10th times 2 to the 10th. And what is 2 to the 10th? Huh? Yeah, approximately 1,000. And so 2 to the 20th is 1,000,000, right? So this is like, if you count, like this is how you kind of, this is how, especially if you're dealing with people outside computer science, right? This is how you make them think that you're really smart. Because you just like come out with a number and they're like, wow, okay. Okay, so you should know all these. 2 to the 10th, and then any power of, okay? So, so if there's 1,000,000 possible sequence of length 10, okay? So the genome is length 3 billion, right? So how many sequences are there? On average, if the genome is random, how many times will you see a sequence of length 10? 3,000 times, right? Because if the genome is random, every sequence will occur on average, right? There's only 1,000,000 possibilities, so you'll see each one about 3,000 times, okay? So now when you're doing your matching, what happens? Well, one of those positions is going to be matched to the right one, but you're going to have about 2,999 other ones that it matches just by chance, okay? So that's not going to work, that method. But it's not a bad idea. That strategy is called sequence assembly, and the original Human Genome Project, that's how they did it. They looked for overlaps, okay? But their sequences, their reads were like length 10,000. So then the random matches were much smaller. But even still, because the genome's not random, there actually were a lot of repetitive regions, and that was actually the main challenge. So if we're not going to use that strategy, what's another strategy that we can use? Yeah? Okay, great. So that strategy is called, so we know that my genome is very close to the human genome. This strategy is called resequencing. So here's the basic idea. Here's my genome. Okay, obviously I don't see the whole thing. And you can see in bold, there are these different positions. Okay, those are the places where there's variation. So let's say I get a read from here. This is just a random sequence. It actually comes from here, right? But of course, I just get this. So what I do is I go to I download the human genome because now it's available. And what I do is I just look for where does this one match closest to the human genome, because I know that my genome is, individuals are only differ by 0.1% of their DNA. So only 1 in 1,000 positions are different on average. So it's going to be pretty close. So there's going to be a position that's going to be really close. Okay. And then what I do is so what do I do next once I have this? How do I figure out what my genome is? once I match to the closest position. Yeah?
You substitute your bases with the bases of the human genome?
Yeah, so I look at where they differ, okay, right? Here's a difference, where else is a difference? Here's a difference, and then here's a difference. So those are those three differences, okay? And what I do is I just record these differences, okay? And I keep on repeating this process, and every time I make a long list of all the differences. And then what I do is I take the human genome sequence and I kind of add these differences to the human genome sequence, right? And then that's going to be what my genome is. Yeah.
Whose genome is the human genome?
Okay, so it's very funny you ask. Okay, the public effort, Francis Collins' led effort. So it was like this, panel of anonymous donors, like representing like different ethnic groups and all this kind of like, the people were decided in a very ethically warm and fuzzy type of way. In Celera, it was a complete mystery who the genome was. And then like a few years later, Craig Venter, he said that it was his genome that they sequenced. So he gave the sample. So it's funny, like people talk about like, people like go and give, analysis of the human genome, and he'll give talks which are like analysis of my genome, right? This kind of thing, right? So, but think of it as like a random person selected from the population, okay? So it's nothing really special. Okay, so does that make sense? So that's, yeah?
Don't you still run the same problem where your sequences show up multiple times?
Yeah, so we'll talk about that. But for at least the unique parts of the genome, it's not going to have a problem. And if remember, your read is now like 30, right? So there's 4 to the 30 different possible full-length reads. That's a much larger number than 4 to the 10, right? So that's actually, 4 to the 30 is much larger than 3 billion. So if the genome is random, you won't have a problem, okay? So, okay, let me just get a little farther because we're a little bit running behind. Oh, we only have 5 minutes left. Can we go to 155 today? It's really good. It's really good. Okay. So why do we need? Okay, fine. I won't do that. Okay. Why do we need computer science? So the sequences are long. The sequencers generate many reads, and we need efficient algorithms to match each read to the genome. And there are other challenges which we're not mentioning. Okay. So what is a simple algorithm? Okay. So here's a simple algorithm to find where you match. Okay. So anyone make a guess? Simple algorithm. Exactly. So here's a read. You basically slide it along. Okay. There's your genome. You try the first position. Okay. Does it match? No. These are all the mismatches. How about the next position? Does it match? No. Next position, match? No. I got bored of PowerPoint, so I skipped a bunch of steps. Does it match? No. Okay, there. It matches. Okay, so we repeat this process. Okay, and so it's a match. Okay, so what's the problem with this algorithm? Yeah, it takes forever. So let's look at how long will it take. The genome's 3 billion, 300 million reads. Reads are like 30. The number of mismatches. And let's say your computer can check a match versus a mismatch in 1 in a million seconds, like a million matches a second. So then the total time will be n times m times l times t, which is like 27, I think, pentillion seconds, which is about 854,000 years. Okay? So that's not going to work. So any ideas on how to speed this up? Yeah? Any ideas? Yeah?
You don't have to make all those comparisons once it's already hit.
Okay, so once you've hit something, then on average you'll hit it in half the time, right? So that'll cut off 400,000 years. So now you're only at like 425,000 years. So any other ideas? Hashing? Someone say hashing? Powerful computer. Yeah. So, yeah. Powerful computer. Yeah.
Only check 5 or like some number for each comparison, and if those are matches, then check the rest.
Yeah. So maybe that'll bring you down to 100,000 years. Yeah. It's progress. Yeah, it's progress. Okay, since I'm running out of time, I'll put you out of your misery. Here's the basic idea, okay? We're looking for a small, like a few mismatches. And so you know that a substring will match perfectly. Here's what I mean. Let's say this is your read, and I split it up into thirds. And let's say I'm only allowing for 2 mismatches, okay? This is kind of the worst case scenario for where the mismatches can fall, okay? What I mean by worst case is that if they fall into different thirds, in either case, you'll always have 1/3 that'll match your read perfectly in the genome, right? So the not worst case would be 2 would match into the same, and then you have 2/3 that would match perfectly. So what I can do is, what I can do is I can basically make an index for the genome, which is like all my sequences in this case of length 10, okay, because that's 1/3 of my read, and I can just record where all the positions in the genome that match those, right? Okay, and on average, how many, for each of these entries, how many of these rows are there? We just talked about it. How many? 1 million. And how many of the entries are there total on this side? 3 billion, right? So on average, there's going to be 3,000 entries. Okay, so so basically, the complexity of this algorithm is going to be like this. We need to look up each third in the index, and so it's going to be there's only going to be 3,000 positions, and for each position, we compute the value, which is going to be. So this algorithm will take about 927 days. So it's about 3 years. So it's a lot of progress. But with other tricks, like for example, if you look for longer reads, it could be brought down to 1 day. Okay? So there's many other problems, and I'm going to actually skip ahead because we don't have time. And I'm going to end with one last thing. Okay? So I want to just mention one thing before we go. So there's actually a bioinformatics minor that's now available if you're interested in this. There's CS courses you could take. And if you're interested in the bioinformatics minor, we have a website for it. And on there, there'll be a lot of information on relevant courses you can take. I teach a course every spring in this topic, and as well as there's other courses. And if you're interested in this, there's a website. You can look it up. And great. So if you guys have any questions, I'll be around, and I'll see you guys later. Thanks.